Hello everyone. In my last video, I discussed in detail about the dihybrid cross of the Mendel which he performed on garden pea plant. In this cross, Mendel considered inheritance pattern of two characters at a time. That means he studied how two characters are transferred from parent to progeny in garden pea plant. Initially, in this cross, Mendel used character like seed color, which is yellow, or green, and seed shape which is round or wrinkle, and then he performed artificial cross in these two parents. And we know that initially the parent yellow and round produce single type of a gamete which is capital Y capital R, and green and wrinkle type of parent produce gamete small y small r, and when they crossed they come together in F1 generation. So the genotype become capital Y small y and capital R small r. So this type of plant always give yellow and round type of a seed because capital Y is present, which is a dominating trait, and small r which is a dominating allele. So that because of this capital Y and capital R, the seed become yellow and round in F1 generation. Then Mendel allow selfing in F1 hybrid, and then he raised F2 generation. Now this F1 hybrid produced four different types of gamete, like capital Y combined with capital R, then capital Y with small r, then small y with capital R, and small y with small r. That means there is equal chance of combination of all possible allele with other type of allele. That means this capital Y can combine with capital R, and this capital Y also go with a small r. Same case for small y. So four different types of gamete produced, each with a 25% probability. And now when these four type of gamete in male and four type of gamete in female fused, this Punnett square give exact number of phenotype and genotype that obtained in F2 generation. And we know that Mendel got phenotypic ratio in F2 generation in dihybrid cross as 9:2, 3:2, 3:1. And in the same cross, Mendel got genotypic ratio 1:2:2:1:4, 1:2:2:1. Okay, I have discussed in detail about the phenotype and genotype which he obtained in dihybrid cross. Now, on the basis of this dihybrid cross result, Mendel proposed a law which is called as third law of inheritance. Okay, so in my last few videos, I discussed monohybrid cross of the Mendel, and on the basis of result of monohybrid cross, Mendel proposed two laws, that is first law of inheritance, also called as law of dominance, then second law of inheritance, which is nothing but the law of segregation. Now, on the basis of results of dihybrid cross, Mendel proposed another law, which is the last one, that is third law of inheritance, also called as law of independent assortment. So in this video, I am going to talk about this third law of inheritance. This law is totally depend upon the finding of dihybrid cross. Okay, so I am taking this law after the dihybrid cross. Okay, so in today's lecture, I am going to talk about third law of inheritance. This law is also called as law of independent assortment. Okay, if we consider the definition of this law, it states that when two pure parents, pure means two homozygous parents differing in two pairs of contrasting character are crossed, the inheritance pattern of one pair is independent of other pair. Okay, the inheritance of one pair is independent of other. Okay, so now if we read this definition carefully, we can understand if we cross two pure parents, two homozygous parents, and each parent differing or have two pairs of different contrasting character, then in F2 generation, the inheritance of one pair of the character is independent of that of the other pair. Okay. For example, in this dihybrid cross, Mendel select two parents. This is the parent generation. First parent have yellow and round seed. Okay, that means seed color is yellow and seed shape is round. Two characters, seed color and seed shape. And in the second parent, the seed color is green while the shape is wrinkle. Okay, so these are the two parents which is homozygous differing in two pairs of contrasting characters. These two pairs are yellow and round and green and wrinkle. Now, if we cross these two parents, then in F2 generation, the inheritance of these two pairs are independent of one another. That means this yellow color can go with round as well as yellow color can pair with wrinkle. Okay, that means this capital Y allele can also go with a round or capital R or small r type of allele. Similarly, this green color or small y gene can combine with a round or wrinkle color. That means in F2 generation, we will going to get four different type of offsprings. One with yellow round seed, another with yellow wrinkle, then green round and green wrinkle. And this prove the law of independent assortment. Okay, so appearance of new combination in F2 generation obviously in dihybrid cross prove the law. Okay, so if we discuss the phenotypic ratio of dihybrid cross, it is 9:3:3:1. Okay, so the ratio is yellow round 9, then yellow wrinkled plant 3, green round 3 and green wrinkle 1. Okay, so we got ratio 9:3:3:1. Out of this four type of a plant, two combinations are original ones. Okay, so this yellow round is a parental combination and green wrinkle is also a parental combination. Okay, these are the two parent combination, but we have got two different combination here, that is yellow wrinkle, yellow combined with wrinkle and green with round. Okay, so these two are the new combinations, and appearance of these two new combination in F2 generation of the dihybrid cross successfully prove the third law of inheritance, which is nothing but the independent assortment law. Okay, what is independent assortment? Two characters are not depend on one another when they transfer from parent to progeny. Assortment is nothing but the distribution. So when this character distribute from F1 generation to F2 generation, they transfer independent of one another. Okay, that means if we consider gamete formation, okay, here this Y can combine with capital R, or the Y can also go with small r, and this small y form gamete with capital R, as well as the small y form gamete with small r. So the four different types of gametes are produced: capital Y, capital Y, small y, capital R, and small y, small r. And production of these four type of gamete is only possible because of independent assortment. And due to the four gamete, 4 into 4, 16 different type of genotype and 4 different type of phenotype appear. Out of these 4 different type of phenotype, 2 were parental combination and the 2 were normal combinations, okay, or new combinations, and appearance of these new 2 combinations prove this law. Another important point that we have to discuss at the time of independent assortment law is that this independent assortment of gene is only possible when genes are located on different chromosome or on the same chromosome but distance apart. That means this law is only applicable to those genes who are not linked. Okay. For example, suppose this is the chromosome with sister chromatid and non-sister chromatid. Now, if one gene is located at this point on the chromosome, then other genes should be located away or distance apart from first gene. Then and then only these two genes can independently assort in the gamete at the time of meiosis. We know that gametes are produced during the meiosis and they are produced by recombination process, and recombination is generally occur in non-linked or the gene which is present distance apart on the chromosome. Okay, so the first condition for independent assortment is that these two genes, capital Y, capital R, or small y, small r, must be present away from one another on the same chromosome, or they must located on different chromosome. Okay, so in the homologous pair of chromosome, the two genes must be present on different chromosome. Then the chances of independent assortment is more. Okay, so we have to remember that when the genes are located away from one another, the chances of independent assortment is high. Okay, if the genes are present close to one another, then these two genes can transfer in the gamete or in the progeny with one another or with simultaneously in a single gamete, and such genes are called as linked gene. Okay, they are linked gene. And linked gene do not show independent assortment. For example, if this yellow color and round seed shape is linked, in that case this yellow color always go with a round seed shape, so that we will not going to get yellow wrinkle type of a seed because this yellow and round is linked. Similarly, if this green and wrinkle is linked, in that case this green color will not going to leave wrinkle gene, so that this green and round combination will not going to appear. Okay, suppose these two genes are linked, in that case in F2 generation we will going to get only two possible combinations, yellow round and green wrinkle. New combinations will not be there. So Mendel was very fortunate because he select 7 pairs of contrasting character in garden pea plant for his study, and luckily all the 7 characters were show non-linkage, or they are not linked. If they are linked, in that case Mendel will not going to conclude anything because of the linkages. The pattern will not going to remain same. The genes will going to transfer with one another, and the ratio may be fluctuate. Okay, so Mendel was very fortunate because all the 7 character which he select was having non-linked genes. Okay, so these are the 2 important conditions for law of independent assortment. Number 1, genes should be located on different chromosome, or if they are located on the same chromosome, they must be away from one another. So this is all about the law of independent assortment. This is the third and final law of the Mendel, which is based upon the result of dihybrid cross. In this law, Mendel proposed that when two pairs of contrasting characters are studied simultaneously in two homozygous pure parents, the inheritance pattern of one character is independent of that of the other character. Thank you.